I'd like to tell you about some of our work about immunity against Omicron. Here are my disclosures. So as I think you know, the US has really led the efficacy trials of 6 different vaccines. The first 2 are the most widely used mRNA vaccines from Pfizer and Moderna. The next 2 are the adenovirus vaccines from J&J and AstraZeneca, and the last 2 are the protein vaccines from Novavax and Sanofi. And it's really quite incredible that 6 out of 6 showed safety and efficacy. It's amazing if you think about it. If you do an experiment, most experiments fail. Here, 6 out of 6 large phase 3 trials all worked. The first 2 of these vaccines are fully approved. The J&J vaccine is approved under EUA, and the last 3 are not yet approved. So in terms of immunity against Omicron, I'd like to review a couple key figures from a paper we published a long time ago, a year and a half ago, which is ancient history in COVID timescales. We published at the end of December of 2020 a study in Macaques on the correlates of protection. We had already shown at that time that antibody titers induced by vaccination correlated with protection, but we wanted to actually prove that antibodies could protect. So we did a classic adoptive transfer study. We took convalescent macaques, we purified IgG, and we did an adoptive transfer study into naive macaques. And we showed that there was a dose-dependent protection. At a high dose, we showed that antibodies alone could block infection. So that's essentially formal proof that antibodies alone can protect. At a mid dose, there was 1 out of 3 infections. At a low dose, all animals were infected, but controlled virus more quickly than controls. So this essentially demonstrated that there was a dose-dependent protection by antibodies against SARS-CoV-2 challenge in macaques. So then we looked at the question of whether CD8 T cells might contribute. And you might say, well, why do we even do this experiment? I just showed you antibodies alone can protect. The key thing is that antibodies protect if they're at a sufficiently high titer. And now in the Omicron era, I would argue that the titer needed to protect against a hypertransmissible variant might be different than the titer needed to protect against a much less transmissible variant. So in this study, which was of course conceived and executed well before the variant era, we wanted to ask the question, do CD8 T cells contribute to protection in the setting of suboptimal antibody titers? So all the details are contained in this paper, but this is the key figure. We took convalescent animals, we let them rest for 2 months, and then we either administered a sham antibody or a CD8 depleting antibody. And then we did a re-challenge of those animals. If we treated with a sham antibody, then these animals were fully protected. So demonstration of natural immunity, which is what we had shown previously. However, if we depleted CD8 T cells, then there was a 5 out of 5 animals showed breakthrough with about 4 logs of virus. So still a lot lower than acute infection in naive animals, but nevertheless significantly higher amounts of virus than sham treated animals, demonstrating that CD8 T cells do contribute to protection in the setting of suboptimal antibody levels. So in this study, we showed that purified IgG protects macaques against SARS-CoV-2 challenge in a dose-dependent fashion. We also showed that CD8 depletion reduced protection against re-challenge in convalescent macaques with waning antibody titers. So we concluded in this study a year and a half ago that antibodies alone can protect, but that cellular immunity likely contributes when antibody titers are borderline or subprotective. And this was largely before we saw the problems of waning immunity with vaccines and before we saw the issues raised by viral variants. But this can largely be interpreted in the modern era. And I think this general concept, this immunologic framework, has largely held intact for the last year and a half. So of course, we're now in the Omicron era. We saw an acute and abrupt change from Delta to Omicron in most countries around mid-December, in some countries in early November, and in virtually all countries by January. And of course, now it's a splintered Omicron lineage. It's not just Omicron BA1, but there's BA1.1, BA2, which is now the dominant virus. We now see BA2.12.1, as well as BA4 and 5 coming up. And of course, what was worrisome about Omicron is the sheer number of mutations, as well as over 30 mutations in spike. substantial increase in mutations compared with Delta. So it was predicted from very early on that there would be substantial neutralization escape with Omicron. And indeed, that was the case. This is some of our data that's actually not yet published, but is actually very similar to data published by at least 24 labs around the world. So this is not novel at all. It just happens to be our data. This is very similar to what many, many groups have reported. This is individuals who received the Pfizer vaccine, and at 6 months, they received a boost. So week 0 is the time frame compared with the boost. And like has been reported before, none of the primary vaccine regimens give much neutralizing titers against Omicron, which is in green. There are still low titers 6 months after vaccination against the vaccine match strain, very low against Delta and Beta, and virtually undetectable against Omicron. But with a boost, 2 weeks after a boost, there's a very high spike in antibody titers, including against Omicron, and these then wane quite a bit, about 8-fold by 4 months later. But 4 months later, we still have low levels of neutralization against Omicron BA1 and BA2. So I think it's fair to say, with the number of real-world efficacy studies that have come out, that vaccine protection against acquisition of infection with Omicron appears to be very modest. And it really doesn't even matter if you have multiple boosts. So even after 3rd and 4th boosts, protection against acquisition of infection is modest with all the vaccines and wanes rapidly. But fortunately, vaccine protection against severe disease has remained largely intact. And the first data that came out, now there's data in the US and the UK and many other countries, but the first data that came out with Omicron and severe disease was from South Africa. So Glenda Gray and colleagues from South Africa demonstrated that vaccine protection against hospitalization and ICU level of care with Omicron in South Africa remained robust, even in the absence of high titers of neutralizing antibodies. And because the 2 vaccines that were rolled out in South Africa were the Pfizer vaccine and the J&J vaccine, both of these vaccines showed very good efficacy. Pfizer was 70%, J&J was 85%. A follow-up large population study that was just published in the New England Journal by the same group last week showed very similar numbers to these initial numbers that were in smaller cohorts, basically protection of both in the mid-70s. So what does this mean scientifically? We're seeing robust protection against severe disease, virtually no protection against or minimal protection against acquisition of infection, largely in the absence of neutralizing antibody titers. And these are 2-shot vaccines, not the 3-shot vaccines that do raise neutralizing titers against Omicron. So we know that there was minimal neutralization against Omicron. So the protection in the absence of neutralizing antibodies would suggest that immune mechanisms other than neutralizing antibodies likely contribute and maybe even are critical for protection against severe disease. So what could these be? These could be antibodies that are Fc functional and non-neutralizing. These could be innate immune responses. And these could also be T cell responses. And so without discounting the potential contribution of those other components, then I'll focus some comments on the role of T cell responses. These are CD8 T cell epitopes that were mapped by Albert Grafoni and Alex Sete against the prior variants in green. The x-axis is the amino acid sequence across the spike protein, and the y-axis is the response frequency. Superposed on this graph are the vertical lines that include all the Omicron mutations. So you can see some of these mutations are predicted to eliminate some of these epitopes, but most of these epitopes remain intact. So theoretically, we would have actually predicted that 80 to 85% of CD8 T cell responses, and similarly for CD4 T cell responses, would be cross-reactive with Omicron. And indeed, that was the case. So we published a paper at the end of January of 2022. The same week we published this paper, at least 3 other groups around the world published very similar papers, and all of the papers came to the same conclusion, which is that all the current vaccines that elicit T cell responses generate highly conserved cellular immunity to Omicron. And it really is in stark contrast with what was seen with antibody responses and Omicron largely escaping antibodies. Omicron does not escape vaccine-induced T cell responses. So here's the data to show that. This is just our data. Again, this is similar to what was simultaneously published by at least 4 other groups worldwide. And here we looked at two vaccines, the J&J vaccine on the left and the Pfizer vaccine on the right. And we looked at 1 month and even 8 months following vaccination. And we showed that 8 months following vaccination, both of these vaccines generated long-term durable CD8 T cell responses, a bit higher with the J&J vaccine than the Pfizer vaccine, but in both cases, highly cross-reactive to Omicron. The vaccine-matched responses are shown in black, and then the cross-reactive responses to Delta are shown in purple and to Omicron are shown in green. So you can see here that the Omicron responses are about 80 to 85% of the Washington AA responses, which is exactly in accordance with theoretical predictions. If we look on a per participant basis, then here are the correlations that we see. These are astoundingly good correlations, given that this is a T cell bioassay in MHC mismatched individuals. And what we see is the vaccine matched CD8 T cell responses on the x-axis and the Delta or Omicron CD8 T cell responses on the y-axis. On the left, we have the J&J vaccine. On the right, we have the Pfizer vaccine. And we see almost a linear correlation with remarkable R and P values. If we look at CD4 T cell responses, then these also are highly cross-reactive. 8 months, 1 month and 8 month following vaccination, we looked at Washington, Delta, and Omicron CD4 T cell responses. And we see that the Omicron CD4 responses for both of these vaccines is almost the same as the vaccine matched Washington response. And if we look at the correlations, we see actually an even better correlation. Here we see a correlation with slopes of 0.8 to 1, which is really quite astounding, given the natural variability of T cell responses. And if we look at other T cell subpopulations, CD8 central memory cells, CD8 effector memory cells, CD4 central memory cells, and CD4 effector memory cells, then we see a similar phenomenon that really whichever time point and whichever subpopulation of T cells we look at, we see a very good level of cross-reactivity. There are occasional responses that appear to fall out, but the vast majority of responses are really quite similar. So to explore Omicron immunity in more detail, we also did a preclinical study in macaques. So we wanted to evaluate the immunogenicity and the cross-reactivity, as well as the protective efficacy and immune correlates of both of these vaccines, the mRNA and the adenovirus vaccines against Omicron challenge in macaques. So in this study, we vaccinated 30 cynomolgus macaques, 6 per group. There were 4 vaccine groups and 1 control group. The 4 vaccine groups received either the Pfizer vaccine as a prime at week 0 and 3. The first group was then boosted with a 3rd shot of Pfizer. The second group was boosted at week 14 with a 3rd shot of J&J. Groups 3 and 4 were primed with the single shot J&J vaccine and were boosted at week 14 with either the Pfizer vaccine or the J&J vaccine. And the last group was a sham control group. We then did a high dose 10 to the 6th PFU SARS-CoV-2 Omicron challenge at week 19, 5 weeks after the last boost by the intranasal and intratracheal routes. This is a study outline just for simplicity. The primes were either at week 0 alone or week 0 and 3 with Pfizer or J&J. And at week 14, the animals were boosted either homologously or heterologously. And then at week 19, the animals were challenged. So if we look at the neutralizing antibody responses, this is prior to challenge. At week 8, this represents after the prime immunization. We see high neutralizing antibody responses against the vaccine-matched Washington strain in black in the 2 Pfizer groups, and about a log lower in the 2 J&J groups. At week 8, cross-reactivity was relatively poor to the Delta and the Omicron strain. At week 14, we see that those high neutralizing antibody titers decline sharply in the 2 Pfizer groups, as we see in humans. The cross-reactive responses decline less strikingly. After the boost at week 18, responses in all the vaccine groups went up tremendously. We saw the highest neutralizing titers in the triple Pfizer group, shown on the left, a lower slightly lower responses in the two heterologous groups and about a log lower neutralizing titers in the J&J times 2 group. In all cases, there was cross-reactivity to Omicron that was about 10-fold lower and intermediate responses to Delta. So pretty much as you would expect, I think. We next looked at CD8 T cell responses. And similar to what we and others have seen in humans, the CD8 T cell responses with the J&J vaccine were higher than with the Pfizer vaccine, and in all cases were highly cross-reactive between Washington Delta and Omicron. After the boost at week 16, the CD8 T cell responses went up, but only marginally, probably with the biggest increase in the Pfizer primed animals that were then boosted with the J&J vaccine. If we look at CD4 T cell responses, these were relatively low at week 14 prior to the boost and went up considerably at week 16 post-boost. The highest CD4 T cell responses were in the triple Pfizer group. Lower CD4 T cell responses were in the two heterologous groups, and the lowest CD4 T cell responses were in the J&J times 2 group. So the CD4 T cell responses tracked largely with antibody responses, and the CD8 T cell responses really looked like an independent parameter. So here are the viral loads in these animals following challenge in bronchoalveolar lavage. Remember, this was a high dose Omicron challenge. So because it's a heterologous virus and a high dose, we would not have expected to see any sterilizing immunity, and we did not. All the animals became infected. And so in effect, all of these animals are essentially breakthrough infections. However, there was a sharp difference in viral loads in the vaccinated animals compared with the sham controls. In the sham controls, there was high viral loads by day 2 at about 5.5 logs. And this then declined sharply by day 7. These viral loads are very similar to what we've seen with prior variants in monkeys. In the 4 different vaccine groups, in all 4 groups, we saw a substantial decline of peak titers, peak viral load titers on on day 1. And in almost all cases, virus was controlled by day 4. So if we quantitate this and we look at peak viral loads on the left and day 4 viral loads on the right, then we can see that all 4 of the vaccine regimens resulted in a statistically significant reduction of peak viral loads. And on day 4, then essentially they were virtually all negative. This study was not really powered to compare different vaccines against each other, but just in terms of point estimates, the 2 heterologous vaccine regimens had the lowest point estimates of viral load, followed by the triple Pfizer, followed by the double J&J. So here are viral loads in nasal swabs. And here we actually saw something quite interesting. First, in the sham controls, we saw that all the animals had virus replication in nasal swabs at about 4 logs. This peak is a bit lower than what we're used to seeing with other variants, but it was sustained over time and only minimally declined by day 7. In the 4 vaccine groups, there was to some extent a variable reduction of peak viral loads on day 1. And most animals had either controlled or almost controlled virus in nasal swabs by day 4. But the protection in nasal swabs was not as striking as the protection in BAL, which is what we would expect. What we did not expect is really to see 4 cases of clear vaccine failure, at least in the upper respiratory tract. 2 animals in the triple Pfizer group and 2 animals in the Pfizer prime J&J boost group. So we had 4 animals that were clearly very different than the other vaccinated animals and looked essentially like sham controls. So the question is, what is different about these 4 animals? I'll get back to that in about 2 slides. If we first look at the statistical analysis of viral loads, at peak, only the 2 heterologous vaccine regimens resulted in a reduction of peak viral loads compared with sham control. The triple Pfizer and the double J&J did not have a significant reduction of peak viral loads in nasal swabs. By day 4, the 2 heterologous groups, as well as the J&J only group, showed a significant reduction of viral loads in nasal swabs, but not the triple Pfizer group, probably driven by these 2 animals that were the high early breakthroughs. We also looked at infectious virus titers by TCID50 assays in both BAL and nasal swabs. So these are TCID50 assays on day 2, and you can see there is a significant reduction of infectious virus titers in both BAL and nasal swabs on day 2. So now we wanted to turn briefly to these 4 animals that essentially were vaccine failures, or at least maybe we should call them upper respiratory tract virologic failures, because they clearly still control virus well in the lower respiratory tract. So what were the immune characteristics of these 4 animals? So here we're showing Omicron-specific neutralizing antibody titers on the left and Omicron-specific CD8 T cell responses on the right prior to challenge. This is the same data that I showed you several slides ago, except I took out all the other time points and I took out all the other variants so we could focus exclusively on the Omicron responses. And we've highlighted in red the animals that did not control virus following challenge in the upper respiratory tract. So clearly that includes the 6 control animals. And also we've highlighted the 2 animals that did not control virus in the triple Pfizer group and the 2 animals that did not control virus in the Pfizer Prime J&J boost group highlighted in red. So where do these fall? These actually have moderately high neutralizing antibody titers, or at least middle of the road. So neutralizing antibody titers of about 1,000, which is pretty high, but middle of this range in this study. And these 4 animals had undetectable CD8 T cell responses. So it appears like the immune characterization of these 4 breakthrough animals involves moderate neutralizing antibody titers and low to undetectable CD8 T cell responses. Well, how about this animal? This animal also has an undetectable CD8 T cell response, but is black, so it means it controlled well. And how about this animal that had much lower neutralizing antibody responses, almost negative, but this animal also controlled well. So keep those 2 animals in mind too. So what we did is we created a 2-dimensional map, and we mapped out the immunological profiles of all 30 animals in the study in essentially 2-dimensional immunologic space. So on the x-axis is the log neutralizing antibody titer against Omicron. On the y-axis is the log CD8 T cell response against Omicron prior to challenge. This is all the animals, all 4 vaccine groups plus the controls. And the animals that did not control virus post-challenge in the upper respiratory tract are shown in red, like the previous slide. The 6 control animals are the ones that are bunched up in the lower left corner, shown here. And the 4 vaccinated animals that also didn't control virus are shown here in red. So we could draw a dotted line to essentially define the immunologic space associated with virologic failure. And this is, of course, a post hoc analysis. And so this should be seen as hypothesis generating and not as conclusive, because this is all post hoc. But nevertheless, it is striking that all the vaccine failures clustered together. And they cluster as essentially low to moderate neutralizing antibody titers and low CD8 T cell responses. So the 2 animals I showed you earlier, this is the animal that had very low CD8 T cell responses, but still controlled. It had extraordinarily high neutralizing antibody titers of over 10,000. It was actually the highest neutralizing antibody titer in the whole group. And as I showed you from the December 2020 paper, if you have high enough neutralizing titers, you don't need T cells. That alone can control virus. Unfortunately, it appears that for Omicron, those titers are largely not attainable in most cases. How about the other animal that had very low CD8 T cell responses, very low neutralizing antibody responses, but still controlled? This animal is here with not the highest, but certainly among the higher levels of CD8 T cell responses. So it really suggests that both humoral and cellular immunity contribute to control of this virus. If we look at correlates of protection in a straightforward analysis, then we find that both neutralizing titers as well as binding antibody titers on the bottom correlate with reduced viral loads at peak and on day 4 here in BAL and not shown here in nasal swabs. And we also show that CD8 T cell responses on the top as well as CD4 T cell responses on the bottom also correlate with reduced viral loads at peak and on day 4 here in BAL and also not shown here in nasal swabs. So what can we say about this study? we observed rapid virologic control in vaccinated macaques in the lower respiratory tract following SARS-CoV-2 Omicron challenge. We also saw significant control by heterologous vaccine regimens in the upper respiratory tract, less good with the homologous vaccine regimens. We show that the Pfizer vaccine induced higher antibody responses, the J&J vaccine induced higher CD8 T cell responses, and the data on the breakthroughs, as well as the correlates, suggest that both antibody and CD8 T cell responses correlate with virologic control, and virologic failure in the upper respiratory tract was associated with moderate neutralizing titers and low to undetectable CD8 T cell responses. So how do these vaccines perform against Omicron in humans? And here are updated CDC data just downloaded directly from their website, showing the performance of all three of the vaccines in the United States over time. And the unvaccinated individuals are shown in black, and the breakthroughs per 100,000 population are shown in the three colors. And as you can see here, all three vaccines provided very good protection. During the Delta wave in September, then there was slightly higher breakthroughs with the J&J vaccine. During the Omicron wave, higher breakthroughs with all of it, but actually relatively slightly lower breakthrough rates with the J&J vaccine and slightly higher breakthrough rates with the Pfizer vaccine. And this is actually similar trends seen if you look at individuals who received only the primary vaccine series without a booster. So what do we conclude in terms of variants and boosts moving forward? I think data from macaques and humans that I've shared with you today suggests that both antibodies and CD8 T cells, and not antibodies alone, are actually important for vaccine protection. Short-term neutralizing antibody responses are clearly important, but I would argue that the durability of antibody responses, as well as CD8 T cell responses, are likely critical for long-term protection, particularly if you look at protection against severe disease with viral variants. Most protection now is really not protection against infection, but protection against severe disease with variants that largely escape antibodies. So in that sense, it's not surprising that CD8 T cell responses might play more of a role in protection in 2022 than it did, say, protection a year ago. Boosts are clearly useful and powerful and currently dominate much discussion. But I would argue the top priority really should remain providing initial vaccines to the unvaccinated individuals in the United States and throughout the world, a topic that seems to have fallen off the agenda. And I would also argue that a strategy of boosting the entire population every 3 to 6 months is probably not a practical or even a desirable strategy for the developed world, and is simply not possible for the developing world. And so vaccine platforms with improved durability would be desirable. I'll stop there. This is credits for our NHP studies at BIDMC and BioQual and multiple other academic institutions, and our clinical trial unit at BIDMC and our industry and academic partners. Thank you all very much. This is a picture of our lab at our retreat in January, and our hope is that we will soon be able to have a retreat in person. Thank you all very much. I'll take any questions.
Thank you, Dan. And I'll just tell everybody that I think Dan has a pressing appointment soon. So if you have any questions, please raise your hand right now. If not, I'm going to go ahead and ask a couple of questions. And the first is, in the animals that you sort of define as failures in terms of vaccination, would you have been able to predict beforehand, knowing their antibody levels and knowing their CD8 T cell levels, that they would have been failures if they fell in that particular region of the graph, for example?
Yeah, that's a really interesting question. So we developed the model. We have a prediction tool now. We developed the tool based on this data set.
Yeah.
So it would not really be appropriate to say, going back in time, we could have predicted it, because we developed it with this data. So we really need a new study to be essentially a validation data set. That's why I wanted to emphasize that what I was presenting really should be viewed as post hoc and hypothesis generating in nature. I would say if these numerical values predict protection in the next study, then we should consider it validated. We're not there yet.
And sort of to your one of the last points that you made regarding trying to get additional people vaccinated, can you comment on how well people or animals are protected against further infection upon an initial infection that doesn't involve vaccination?
Yeah, I think it's been very controversial And I'll remind you that our very first paper on SARS-CoV-2 that came out, I think in May of 2020, was a demonstration of natural immunity in monkeys. Our first experiment actually was showing that if we infect monkeys and we re-challenge them, they're largely protected. I think that natural immunity was not appreciated very much in the earlier days, because with the mRNA vaccines in particular, the antibody titers are so high 2 and 4 weeks out that they sort of dwarf the antibody titers of natural immunity. But what's been seen is that those high titers with mRNA decline quickly, and the titers with natural infection kind of stay largely intact. So after a point in time, then there's not much of a difference anymore. And I think there's much more appreciation now of the importance of natural immunity and protection. And I think that's seen very clearly in population cohorts throughout the world. So I forgot your actual question, but I think your question is in the absence of vaccination, how well are you protected? Yes. So it turns out you're protected pretty well. It looks like natural infection gives you about as much protection as vaccination. It used to be thought of as much less, but I think that the pendulum has swung more to being relatively similar. Now, I'm not advocating people to get infected. It's much better to be vaccinated. But if you are infected and you were not vaccinated, then it's probably relatively similar. At least that's what
I guess what I'm asking is, in terms of policy decisions, is it reasonable to say that a person who's not vaccinated but has been infected, there's perhaps no need that they get a vaccination? I think this is how it's treated in Europe.
Yeah.
And I'm just curious whether what are your thoughts on that?
Well, it's sort of any policy is politically charged. And from a science perspective, that sounds reasonable to me. But I would say that if you're naturally infected and if you choose to get vaccinated, then you will have immune responses that go through the roof, and you will be very, very well protected. So you actually still will have a lot to gain by getting vaccinated.
Okay. We have a question from Janine Diarmento. Janine, you can ask your question.
Hi, can you hear me? Yeah, I guess my greatest concern about this is that kids who are vaccinated and then infected, university kids are being asked to boost, mandated to boost by universities. And it seems like that would be a policy decision that doesn't make sense if there is some protection given by the infection after the vaccine, and maybe they can stop these mandates.
I'm not a policymaker, and I do apologize. I'm going to have to go in less than a minute. I have to introduce someone for a seminar, actually. But I'll just say that I'm not a policymaker, but I don't see those policies as science-based.